Hi, I'm Deborah Shera, and I'm in Dr. Tim Gavin's lab at Purdue University. And today I'll be presenting on skeletal muscle extracellular vesicle regulation of angiogenesis is dependent on fiber type. Skeletal muscle EV signaling is an emerging field of study. However, there's little information known regarding how skeletal muscle EVs from different muscle fiber types regulate the surrounding capillaries. Skeletal muscle regulates endothelial cells via the secretion of angiogenic signaling factors, including extracellular vesicles. So angiogenesis is the development of new blood vessels. Muscle can be characterized as predominantly glycolytic or oxidative. So glycolytic is white muscle, it's explosive, fast twitch, ATP through glycolysis is done. And then for oxidative, that's red muscle, it's more for endurance, slow twitch, and it has lots of mitochondria. Oxidative muscle is more capillarized and generally secretes more traditionally pro-angiogenic factors, notably VEGF. So VEGF is the primary angiogenesis factor, then glycolytic muscle. And previous research indicates that muscle EVs may contribute to angiogenesis. It is unknown if muscle fiber type regulates EV angiogenic potential, so the aim of this project is to test if the pro-angiogenic properties of muscle EVs are different between oxidative and glycolytic muscle. And if this is the case, this is a potential mechanism that could contribute to oxidative muscle having more capillaries than glycolytic muscle, so it would explain that. For methodology, oxidative muscle was isolated from the soleus and gastroc in male mice, and glycolytic muscle was isolated from the quadriceps. EVs were isolated using ultrafiltration-size exclusion chromatography, and EV number was measured using NanoSight. HUVECs, human umbilical vein endothelial cells, were treated with OXI-EV or glycolytic EV for 24 hours, and then the functional angiogenic responses and molecular pathways related to angiogenesis were analyzed via Western blotting and PCR. So you can see up on this table, up on the top, this is just a nice visual representation of our methodology. And then differences between groups were determined using paired student t-tests or repeated measures ANOVA. And then you can see on the bottom, we have the red versus the white muscle. So we could see from the data that we got that oxidative muscle released more EVs than glycolytic muscle. So you can see that on the top graph, top right. And then in HUVECs, oxidative EVs promoted greater tube formation, lowered intracellular superoxide and overall reactive oxygen species, and increased cell migration compared to glycolytic EVs. So we have the ROS graph here, migration, number of tubes, and tube length, and then we have the oxidative and glycolytic EVs. So we can see that oxidative muscle had the greatest functional response. Treatment with oxidative EVs increased HUVEC eNOS phosphorylation and gene expression compared to glycolytic EVs. So eNOS creates nitric oxide, which vasodilates the blood vessels. This increases blood flow and provides more oxygen. Gene expression of VEGF-A and FLT1 increased in OXI-EVs compared to glycolytic EVs. There was no difference in expression of FLK1, Ang1, Tie2, or VE-cadherin between oxidative and glycolytic EVs. And then following LNAME treatment, LNAME is a nitric oxide synthase inhibitor, so it suppresses nitric oxide production. Following this treatment, there was no differences in HUVEC tube formation, overall ROS, or cell counts between oxidative and glycolytic EV-treated cells. So looking at this data, EVs likely contribute to the stronger pro-angiogenic signaling properties of oxidative muscle fibers, and this is consistent with the greater muscle capillarization. Capillary differences in muscle may be regulated not only by just the greater EV release, but also the greater EV angiogenic potential from the oxidative muscle fibers. And so oxidative muscle EVs elicited a stronger pro-angiogenic response via the modulation of the nitric oxide signaling pathways. So looking at the nitric oxide, vasodilation of the blood vessels, increased blood flow, more oxygen. Some limitations to this are that the cell culture was not a perfect model of in vivo, so there was no hypoxia and there was no blood flow component, so those are just some things to keep in mind. And then for future investigations, looking at how do various disease states impact muscle EVs, or how does exercise impact EVs between fiber types? And also how do EVs in highly trained athletes, so like an endurance athlete, compare to those of sedentary individuals? And these are my references. So thank you so much for your time. Thank you.